evolutionary biologist, so I have to start with Charles Darwin. Probably you have seen it quite a bit in this lecture series. But it does make a lot of sense when it comes to my research topic. And one of the reasons is that I'm actually interested in one of the central questions still about evolutionary biology, and that is still the origin of species. Here what you see is the cover, the original print of Charles Darwin's seminal book. And you see that here really what was sort of printed in the largest letters, in the most capital letters, if you want to put it like that, is the origin of species. In fact, many evolutionary biologists say that what Darwin actually did not answer was how, in fact, new species evolve in the sense of the meaning of the actual process. So his book is a lot about natural selection, of course, adaptation and a lot of mechanisms. But how exactly out of one ancestor two or even more species evolve is, well, still something that keeps us busy after, well, many years of research. One thing that I would like to point out here, that he actually started out his career with some field work, some extensive period of field work, I should say. In fact, he was, as probably all of you know, 5 years, or about 5 years, on board of the so-called HMS Beagle, this ship, as a very young scholar. And I think a lot of the experiences that he underwent on this trip, on this journey, is what made him actually see things. And you see here that this journey, I would say, literally took him around the world, and he visited a lot of places, saw many different things, collected a hell load of samples, in the end grabbed each and every creature that he could get his hands on. But of course there is also this one spot, and this is of course the Galapagos archipelago. It's in our collective minds for many reasons. In fact, maybe it shouldn't be so, for the reason that these birds that Darwin saw there, this is in fact original drawings, were most likely not as influential to the formulation of his theory as it's commonly thought. He gave the birds to some bird specialist and didn't consider them much. But there's still one thing about these birds, and I think this is why I would like to mention them, that these are the classical textbook example of this exact process of adaptive radiation. Adaptive radiation is nothing else than the evolution of a great deal of ecological and especially morphological diversity within a short period of time and from a common ancestor, and as a consequence of the adaptation of the new species to distinct ecological or environmental niches. And this is so nicely illustrated in these Darwin finches, and that's why they are such an important model system. It's of course nicely illustrated because one can immediately see how that adaptation works and what is the phenotype, the morphology, that actually makes this adaptation. And this phenotype is clearly the beaks of these 13 or 14 finch species. So what you see here is what is called a phylogenetic tree, so a branching diagram that tells us the relationships between the different species, in this case made on molecules or based on molecules, so using DNA and constructing such a phylogenetic tree. And what you see here are the different species, and in these cartoons you see the different beaks, and of course in this case we know in these systems what they are used for, and it's rather obvious how adaptation works in that system, in the sense that some of the species with these sort of large beaks and strong beaks, they are the ones that can really sort of break the big seeds, whereas those ones with maybe more pointed beaks and so on, they are specialized to eat smaller seeds out of cactuses, for example, and so on. So we have this adaptation, and very importantly, this clear correlation between a phenotype that is adaptive and, in this case, the actual food source that the different finch species feed upon. And there's two things that are important about adaptive radiation. One is that a lot of people think, including myself, that a great deal of the morphological diversity on Earth emerged via such adaptive radiations in many different groups at many different times. So it's really a pump of diversity, if we want to put it like that. But at the same time, such instances of adaptive radiation allow us to have a really close look on some of the key processes of evolution. And it especially allows us, so if we look in these diverse groups, in these rapidly radiating groups, it allows us to address questions such as why is there some lineages that have not diversified over long periods of times, whereas we do have, on the other hand, those groups and lineages that exploded literally. So that produced a great deal of ecological or morphological diversity within a short period of time. And this is in fact the question that interests me most. Why is there this difference? Now at the same time, these rapidly radiating groups and these highly diverse groups, these allow us really to address how does, for example, adaptation work, how does innovation work, so evolutionary innovation in the sense that a new phenotype, a new trait or something that is adaptive evolve. And of course it allows us an insight into the very process of diversification or speciation in Darwinian sense. Now the reason why I call it diversification here and not speciation is simply that speciation implies a result, it implies an outcome. So it implies that in the end we have two different species, but in young radiations or when the process is still ongoing, we see that there is some diversification, but in fact we don't know, if we look at it right now, whether it actually leads to two completely ready species in the sense that they are not interbreeding. But what I mean here is, of course, speciation in most cases. And we are particularly interested in sort of the molecular basis behind these processes. So how is that written in the genome and everything that has to do with the genome, so to speak. And the main study group are these cichlid fishes. I always like to quote here George Barlow, who spent basically all his scientific career to work with these cichlid fishes. And he said that he has actually never met something that wasn't interesting. But well, there are obviously some groups that are particularly interesting, that stick out, and he says this is definitively where the cichlid fishes belong. And I think what he had in mind was clearly the diversity of cichlid fish that one can observe. If you're a fan of Monty Python, you know that this could be Wanda, for example. You might have seen some others, like the discus fish in some aquaria and so on. But I guess you don't need to be a fish specialist, an ichthyologist, to see that there is quite some diversity in that group. We have these differences, these great differences in body morphology from exactly these extremely rounded forms like the discus fish to, on the other hand, quite elongated fish like this Julidochromis from Lake Tanganyika. We obviously do have a great difference between the species in coloration and pigmentation traits, colors, stripes, bars, and whatever. And we also do have quite some differences in size. So from this tiny little species in Lake Tanganyika that lives in empty shells that they use as a house. On the other hand, you have quite substantial fish like this guabote in Nicaragua. But I sort of need to tell you a bit more now that I say Tanganyika, Nicaragua, and a few things like that. So I should tell you where these cichlids actually occur and what they are. Cichlid fishes are a tropical and subtropical group of freshwater fishes. They are distributed in the southern continents, and that has most likely a reason that all the southern land masses used to form what is called Gondwana land, so a supercontinent. And the current understanding is that the ancestor of the cichlid fishes sort of already lived on this supercontinent, and so the different lineages got drifted apart with the continents. And then they underwent such adaptive radiations on these different land masses, so that we now find some relatively species poor lineages in India, Sri Lanka, and Madagascar, and then two massive radiations, one in Central and South America, and the other one in Africa, and there especially in East Africa, and that's where we're going now, more precisely to Lake Tanganyika, part of the three Great Lakes in the East African Rift Valley. And these three Great Lakes you see here are aligned, sort of this geological formation, and they are famous for their cichlid fishes for the main reason that there's many of them. So in fact it's estimated that in Lake Victoria there's about 500 different species of cichlid fishes. Lake Tanganyika probably houses about 250 species, and Lake Malawi is estimated to house about 1,000 different cichlid species. Now this is impressive, this is massive. But this is even more impressive if you think that all the species in Lake Victoria only occur in Lake Victoria, and they have actually evolved there. And the same is true for all these different cichlids in the different lakes. So these are unique species. And the second thing why it's impressive is simply the geological time frame in which this happened, because to produce this vast amount of species, it didn't need more than 10 million years in the case of Tanganyika, most likely less, and a maximum of 100,000 years, most likely less, in Lake Victoria. So really rapid speciation and the production of a great deal of ecological and morphological diversity, as we will see here. And again, I would like to point out that we have this sort of two levels of diversity in the cichlid fishes. And one level is clearly the colorational diversity. That's where the German name Buntbarsche comes from. So that's how they are called in German, because they are simply very colorful. And what you see here is 9 male representatives of cichlid fishes from Lake Victoria. And again, I don't think you need to be a fish specialist to see that these are different. Now, knowing that the females of those are, in terms of their coloration, they're a bit more dull, silverish, and don't show that color pattern, and or these color patterns. And this is very often, of course, sort of the case when you have sexual selection via female choice going on. So the females choose the males according to their color, and this is actually happening in this group of fishes. So we have this color diversity. But color doesn't really make an adaptive radiation. So what we have as a second major layer of diversification in these cichlid fishes is really the ecomorphology, or the differences in what is called the trophic apparatus, which is nothing else than a word that circumscribes everything that has to do with the mouth and the feeding apparatus. So just like with the beak of the finches, we do have here differences in the mouth morphology, but in fact we do have many more differences than we do have in the Darwin's finches. So for example, we have these fishes that are insectivorous, and they have tiny little teeth which they use to grab out insect larvae and stuff from the ground. We do have all kinds of zooplankton eaters that have pipette-like mouths so that they suck in water creatures, small water creatures. We have all different kinds of algae scrapers that are specialized to eat algae. We do have all different kinds of piscivorous fish, so fish eaters. Some of them are really torpedo-shaped and they have big mouths so that they can catch other cichlid fishes. We have leaf eaters that eat leaves that fall into the lakes. We have mud diggers and mud shovelers that filter feed stuff that they sort of take up from the ground. Then we have fin biters that are specialized to eat fins of other cichlid fishes. We do have eye suckers that suck out eyes of other cichlid fishes. And we do have even scale eaters. These are probably the most amazing creatures in the sense that they exclusively feed on scales of other cichlid fishes. Who had ever thought that this is enough in terms of nutrition? But it obviously is, because they only eat scales. We know them because we open them. And these are the ones that also have a fluctuating asymmetry, in the sense that about half of the population has a mouth that opens to the left and the other half to the right. The reason is that they can attack sort of the prey fish from the back, from one side. And the idea is that if there's too many with a left opening mouth, natural selection balances them back, because of course the prey fish tend to look more on that side, and knowing that there is an attack. At least that's the idea behind that system. Now again, I have this map here, and basically all this diversity, or a great deal of diversity, has evolved several times in several of these lakes. So what we do have in these cichlids is a massive degree of convergent evolution. Now what I show you here are 5 representatives on the left side of Lake Tanganyika, and here to the right 5 representatives of Lake Malawi. And now you need to be a fish specialist to know the difference, for the simple reason that whatever we see here as similarity is convergence. It evolved several times repeatedly. The fish from Lake Malawi only evolved within Lake Malawi. They go back to one or a few ancestors. But what that really means is that this guy here is much more closely related to this one, to that one, to that one, and to that one, so they share a common ancestor, than it is to this one. The same is true for all of these pairs. So we have evolution repeating itself. But I will come back to that in a few minutes. I have to go back first to this chart, because I was not telling you everything. If you look at cichlids, the mouth apparatus is not only the structures that you see from the outside. Cichlid fishes have something that is called an evolutionary innovation. An innovation in that sense is a trait that enables adaptive radiation. And maybe the most important innovation of the cichlid fishes, which they share with other fishes as well, is what is called the pharyngeal jaw apparatus. And the pharyngeal jaw apparatus refers to an anatomical structure that is sort of hidden in the pharynx, so inside here, that's pharyngeal jaws, and in fact, that's a second set of jaws with a different developmental origin. And this second set of jaws is functionally decoupled from the oral jaws, meaning that it's completely independent, and hence it opens more degrees of freedom to adapt to distinct ecological niches and food types. In order to understand that, we have to have a closer look into the cichlids' mouths. So what we have here is the neurocranium, so that's sort of the skull, if you want to put it like that, that has the brain inside and of course the eyes and so on. And directly below is this, what is shown here in purple structure, that is the pharyngeal jaws, which is a modification from one of the gill arches that are sort of fused to a lower part and an upper part to form an effective mechanism for chewing. So we have here the oral jaws that are then used for biting and catching the prey, and this apparatus with the muscles here is used for chewing. And in fact, it looks a bit like this. So this is a computer tomography of the cichlid. So we have this lower plate and this upper plate, and we really have teeth on it, plenty of them in this case, and you have to imagine all food goes through here, and they actually use that to chew it. So that looks more like a T-Rex than a fish, I would say. But in fact, that's the very effective apparatus to sort of process the food, not to catch it, not to sort of grasp it or whatever, but to chew it. And if we now look at different cichlids, what we observe is that we have a great deal of diversity in the pharyngeal jaw apparatus as well. So these are real fish heads now, and correspondingly we have different pharyngeal jaws. And here we are pretty much where we are at the Darwin's finches, because intuitively we can see what they are eating. So we have even a closer resemblance, and I will show that to you that in a second, between sort of the morphology, the phenotype, and the environment, in this case of course the food that the different species eat. Now what I would like to show you here is simply one of the approaches that we take to understand such an adaptive radiation. In this case, it's a bit more a descriptive approach. What we use is a method called geometric morphometrics. In fact, what we do is we want to compare the morphology between species. Now you can come here and say, well, they are different or they are similar. We want to do it a bit in more detail. So what we use is digital images of fish, and we set what is called landmarks, which is these dots basically here, what is called homologous positions, on positions that we can identify, like the onset of the fins and a few other things. And then we do that with a whole bunch of specimens per species, and then we can calculate difference between species or calculate the variance of the morphology within a species based on a bit more statistically sound methods than just saying they're similar or different. So what you see here, we can do that also with the pharyngeal jaw apparatus. That means we can set landmarks there as well. And of course we need other measurements, because what we want to do in this case is to compare it to ecology. Now again, you could go and say, well, this one eats this and this one eats that by observing or whatsoever. That's not very accurate. We could also open the fish, which we've actually done of course, and look what they have in there. But then what you see is, well, what they've just eaten in the last few hours or so. It's also not very accurate. So what we use is chemical signatures. We can say this species or this individual in the end over the last several months has mainly been eating plants, because this chemical signature tells us that it looks like plant material. It's very important to know evolutionary history, phylogenetic relationships in order to interpret evolution. And this is the wonderful phylogeny of cichlid fishes in Lake Tanganyika. And this, I think, nicely illustrates a species explosion. So this is an adaptive radiation, where really the different species explode. The message of these slides and the colors is that, just for the next slide, remember, the same color means they belong together to a group, they belong or they form a clade, meaning that they are closely related. So same color is closely related, in this case this has even names. Different colors means distantly related. We now look at similarities of this pharyngeal jaw structure, and this is now simply a cluster analysis, so a similarity analysis of the pharyngeal jaws. Then what we see is a rainbow pattern. Basically what that tells us is that the morphological similarity is not explained by phylogenetics. It's pretty much random. In fact, this is a bit too small to see, so I would like to zoom in a bit and show you one representative. So all this group that is morphologically similar, they all look like this, with big teeth and spiny teeth, so these are obviously of a certain foot type. In here you have those ones with the big sort of molariform teeth. Here are these with the very tiny little teeth that I already told you are algae eaters and so on. So the situation that we have here is clearly that the morphology is decoupled from the evolutionary history. Of course it's related to what the different species are eating. So if we now plot, and don't worry, it's not getting more complicated than then, but if we plot, like in this case, the nitrogen signature that tells us where in the food chain a certain species sits, we clearly see that those ones that are morphologically similar also have very similar signatures in the stable isotopes, and this is of course statistically very strongly supported in this case. And so what we can show with this is several things. One that is very important for adaptive radiation, that we do have, of course, also here this tight connection between the functional phenotype, so this jaw structure, and the food type, just like the beak of the Darwin's finches. So this is an important prerequisite, in fact, even for an adaptive radiation. But the second thing goes a bit beyond, because if we now interpret phylogeny into that, then what we actually show is that we have a lot of convergent evolution in this system. For the simple reason that if distantly related species show sort of similar or even the same phenotype, then this is exactly convergent evolution, in case if there is also closely related species that have distinct phenotypes, and both is what we see here, because we see the pink scattered around, and we have here pink next to red, green next to brown, next to blue, and so on. So this is convergent evolution in a very technical way of illustrating it. Let me tell you two more stories here. And the first is really sort of the most extreme case of convergent evolution, but now within an adaptive radiation. And I think this is the first time one has really shown that it can get that extreme. So what we see here are two different fishes, two different species in fact, but they are very similar. I think you agree with me, after I've shown you all these different forms and different colors and shapes before. They are very similar. They are similar to a degree that even local fishermen in East Africa, they think it's the same species for the simple reason that they also catch it on the same sort of hooks, in the same nets, in the same place. But they are distinct. And if you have a closer look, well, this has larger scales than this one and so on. So a fish specialist will see the differences. One is called Ctenochromis pendicola, which doesn't add much. The other has a different name. But look at the colors. I was telling you, different colors, different phylogenetic backgrounds. And if we look at the phylogeny, they are pretty much on the other end of the phylogenetic spectrum. So they are genetically as distinct as you can be within Lake Tanganyika. They are more distinct than human and chimps are in terms of their DNA, which is not much, but in terms of cichlids, that's massive, because there's hundreds of species that are more closely related. But they have undistinguishable pharyngeal jaws. And if you look in their stomachs, you will find in both of them these tiny little pincers of endemic freshwater shrimps of Lake Tanganyika. So they eat the same. What happened is that out of different genetic backgrounds, sort of two lineages literally converge into the same ecological niche, where they feed on the same thing. That's probably why natural selection made them sort of look very similar, and they coexist almost peacefully in this niche. This is by far not the only example that we have. We discovered plenty of such convergent phenotypes within a single lake, within a single adaptive radiation. Of course, if you speak of convergence within the same system, then one thing that you cannot exclude is what is called gene flow. So you're in the same lake. We are claiming convergence, but you cannot exclude that it's made with the same genes. So what we also did, we went even further. And in this study, we looked at this funny and weird thick-lipped phenotype that you find basically in all the cichlid adaptive radiations, also in Central America. And so we went and compared an African and Central American fish with the same methods, even including more genetics and so on, for the simple reason that you can basically exclude gene flow across these two geographic areas. And if you remember back, I told you that has to do with continental drift. That's too old to retain similar genes or something like that. And if we do so, we basically have the same sort of results that if we do our morphometrics again, then we see that in both cases, compared to the ancestor, the snout gets more pointed, so we have the same morphological trajectories in these both cases. And again, it's linked to the same food type, in this case, to hard-shelled invertebrates. So both thick-lipped forms have a large fraction of hard-shelled prey, whereas their sister species or their sister groups eat something very different. We actually found that the same set of genes were expressed in these tissues, meaning that convergence is based on a similar set of genes. And I would maybe like to summarize. So I was speaking about diversification, and I hope I could sort of show you or demonstrate you with my slides that convergence was the outcome of convergent evolution within an adaptive radiation happens, and this could be interpreted as one of the sort of motors of this diversity in Africa, because in these lakes, if we have these many convergent phenotypes that come out of different corners, this could explain coexistence of so many different species in the lakes, that they share niches. I was more speaking about convergence, showing that across different continents even, so without hybridization or anything, similar phenotypes evolve associated to the same habitats. And I was also showing you that there's this innovation in cichlids, this putative key innovation in form of this highly variable pharyngeal jaw apparatus that sort of promotes adaptation to distinct niches, and that clearly reflects, just as the Darwin's finches speaks, the different habitats and food sources that the different cichlid species use. And with this, I would like to thank you very much for your attention and finish with a mask from the Congo.